The other combination I think that is important to mention is the combination of lenalidomide with rituximab. This is the study that was studied at Cornell. And in this ASH 2018, so it's 5 years follow-up. So these are patients that receive induction with lenalidomide and rituximab, and then they continue on lenalidomide. And some patients can stop treatment, I think, after 2 or 3 years. But the important data, at least to me, is that they are seeing some molecular responses in patients treated what we call non-conventional chemotherapy. So if 20 years ago you will tell me that mantle cell lymphoma will be ever treated with just non-conventional chemotherapy, we say, no, it's not going to happen. Well, it's happening. So there is a lot of, in my view, a lot of hope about this combination, rational combination of lenalidomide rituximab, rational combination of ibrutinib with venetoclax as a front line for mantle cell lymphoma. There is still going to be a role for ibrutinib with chemoimmunotherapy for selected group of patients. I think during most of my discussion, I talk about the elderly patients because that's where we are going to see mantle cell lymphoma. But there are patients that are 40 years old, 50 years old, 60s that are going to be diagnosed with mantle cell lymphoma. And in those patients, yes, I want to be more aggressive, trying to induce the larger duration of response, because then I'm going to have time until the new discoveries. If the patient relapses, then I'm going to have more treatments to offer to these patients.
How about this? I am very excited about the potential for CAR T-cell therapy in mantle cell lymphoma. There are ongoing trials both within the Celgene Juno group and within the Kite Gilead group asking this question, can we improve outcomes? Given what we've seen in acute lymphoblastic leukemia, given what we've seen in diffuse large B-cell lymphoma, very encouraged, very excited. I think that we are starting to optimize the management of toxicity, so the fact that these patients are older coming into this therapy is less of a concern for me. I think that we have a real potential here, but we have to see what the trial data ultimately shows.
In terms of the new generation of anti-CD20 antibodies, yes, some of them are more potent than the others. So I think that monoclonal antibodies are going to still be part of the armamentarium, are going to add some value in some scenarios. The example is Michael 1 study, anti-CD20, although it was rituximab, add benefit to ibrutinib in patients with low proliferation rate. Are the new antibodies going to improve the outcomes in patients with mantle cell lymphoma with high proliferation rate? Remains to be seen, right? But that will be, if someone is able to show that the new generation of anti-CD20s increase the efficacy of ibrutinib for any other target agent in this population with high proliferation index, then I would say yes, there is a benefit. There is perhaps because of the different binding to a different epitopes, this new anti-CD20 is improving the efficacy in combination with this drug. But at this point, I think that there is, so we need to test that in a context of clinical trials.
In the maintenance setting, well, what we do know about is monoclonal antibodies. What we don't know about is how we might use those same maintenance monoclonal antibodies if we were able to get a deep response with other agents such as ibrutinib or venetoclax. What I mean by that is that maintenance in that setting might allow you to discontinue the small molecule drugs and reinitiate them at relapse.